Hey everybody! In this video we are going to talk about Beadle and Tatum and their one gene, one enzyme hypothesis. So to start, let me just discuss a little bit about what we think scientists were thinking in the 1930s, 1940s. So most of them were pretty sure that genes controlled traits like eye color and wing shape and height, plant height, size of the animal. But, they weren't so sure about metabolism. So did genes control or they weren't sure if genes controlled metabolism. So do genes control metabolism? And Beadle and Tatum set out to test that or investigate if genes controlled metabolism. And what do we mean by metabolism? So essentially, how do simple compounds or elements like carbon and nitrogen and let's say compounds like water, very simple things, get incorporated into complex compounds. And this can be sort of a cycle, right? And the process of going from a simple compound to a complex compound would be anabolism, and the process of breaking down a complex compound into simple components would be catabolism. And these are both forms of metabolism. And some examples of complex compounds. Well, we talk a lot about these in this course, so amino acids and nucleotides. Just some examples. And we can include vitamins in here too. Why not? So, I'm not really sure what the hypothesis was about what controlled metabolism in cells. If it wasn't genes, what was it? I'm not really sure. But scientists they weren't convinced that genes could control metabolism. So Beadle and Tatum set out to test their hypothesis or investigate if genes did indeed control metabolism, and they worked with a fungus called Neurospora crassa. Neurospora crassa. And this is a fungus my lab uses. So briefly, this is a fungus. It is haploid, meaning it only has one set of chromosomes. So humans are diploid. This fungus is haploid. It has one set of chromosomes. It has 7 chromosomes in that one set, and it is a prototroph. So Neurospora crassa outside in nature, it's a prototroph. And this is an interesting fungus. If there are forest fires, generally the fungus grows on the burnt wood after a forest fire. So if you want to collect Neurospora crassa, find a place where there's been a forest fire, and you should see Neurospora start growing from the trees. I've never seen it. I've wanted to go out and collect Neurospora from the wild, but here in Illinois, we don't typically get too many forest fires, right? We don't have too many forests. So okay, so this is Neurospora crassa, a fungus. And now, since Beadle and Tatum studied this, scientists around the world use this as sort of a model organism, a model eukaryote, right? Fungi are eukaryotes, and they use this fungus to study all sorts of processes that eukaryotes use, or eukaryotic cells use. My own lab uses this organism, and we study a process called meiotic drive, and also chromosome dynamics, particularly during meiosis. And we also study now heterologous gene expression. So being able to take a gene from one organism, put it in another, and get that gene expressed. So we're studying all these different things in my own laboratory here at Illinois State University. So that's an aside. Don't need to worry about that. I should say this is 2020 in case we use this video in the future. This is what my lab studies in spring 2020. So, okay, so what did Beadle and Tatum do with this fungus? Beadle and Tatum, the names of the scientists who were studying this fungus, they did is they took Canidia. Now, Canidia are spores of the fungus, and they're produced asexually. So remember all that meiosis and sexual reproduction and gametes? That's not involved in making Canidia. These are essentially little pieces of the fungus in their spores, and they can be carried, blown by wind, or carried by insects to new locations. And the fungus makes millions, billions. I guess it could make billions, depending on how big of a culture it made. But it makes tons of spores. Each culture can make many, many, many of these Canidia, which is just another name for a spore. So what did they do? They mutated the Canidia with X-rays. And they got lots of mutants, so lots of isolates that had their DNA mutated by X-rays. And they took the mutants and they tried to isolate auxotrophic strains from the mutants. So here's one mutant. And let's say they grew it on complete medium. And this is a test tube. And this is a common way to study Neurospora crassa. Even our textbook kind of shows a test tube with the medium at a slant like this. This is a solid medium. And what you do is you pour the molten medium into the test tube, and then you tilt the test tube at an angle while the medium dries. And so you get this big surface area, because the fungus grows better when it has a large surface area. And so they take the mutants, and let's say they take one mutant, and they grew it on, we'll call this a slant, a test tube slant containing complete medium. And complete medium, as you might remember from previous lectures, contains amino acids. I'll probably get a new pencil. Vitamins, nucleic acids or nucleotides. So anything the fungus might need to grow on medium. You know, the fungus can normally make all these things itself because it's a prototroph, but it's a mutant, and it might be mutated in a gene required to make one of these things. And that's what Beadle and Tatum are trying to find out. Are genes required to make these things? So they grew it on complete medium, and let's say this one right here, it grew fine on complete medium, this mutant, but it didn't grow on minimal medium. Minimal medium. And if you remember from previous courses, this is just a very basic medium containing, say, a sugar, like glucose, salts, minerals, and water. Okay, and it will contain some sort of gelatin too, also, to make the media solid. You don't need to worry about that though. And see, I showed growing here. That was wrong. I shouldn't have done that. The mutant doesn't grow on minimal medium. So they added, they tried the fungus, the mutant strain, on, let's call this minimal medium plus vitamins, plus like every vitamin known. Let's say that. So they were able to use every vitamin known. And the fungus did not grow. Okay. Now, how about, let's see, what else could we test? Nucleotides. So minimal medium plus, the 4 nitrogenous bases, are added to the medium, or even nucleotides. not just the bases, maybe the whole nucleotide, you can add that in there. Essentially, it's not going to grow. It didn't grow. At least this strain that we're using for an example did not grow. And okay, what else could we use? How about amino acids? Okay, we can add all 20 amino acids to the media. And oh, okay, it grew. Okay, so this mutant right here, it said they isolated hundreds and hundreds of mutants. And this one right here grows on complete medium and also on the minimal medium if you add all 20 amino acids. So we can test this further, right? So how would you be able to tell, if the fungus requires all 20 amino acids, if it requires 2 of the amino acids, if it requires just 1 of the amino acids? Well, then you could do 20 more tests, right? 20 more tests with the minimal medium plus 1 of the amino acids, 1 at a time, and see if the fungus can grow when supplemented with a single amino acid. And so that's what they did. And let's just do 2 examples. Let's say plus alanine. So minimal medium plus alanine, the fungus doesn't grow. And okay, let's say minimal medium plus leucine, another amino acid. Let's say this time it grows. Okay, that's pretty good. Now what about the other 18? Okay, it doesn't grow. Minimal medium plus 1 of 18 others, right? Because there are 20 amino acids. And they do this experiment 18 more times with each of the other amino acids, does not grow. Okay, so that mutant requires leucine. Okay, so pretty good experiment, right? So they used X-rays to damage the DNA inside the conidia. Damaging the DNA created a mutant strain, and that mutant strain was no longer a prototroph, was an auxotroph, and it required leucine. So this was the strongest evidence to date that genes control metabolism. And they use this data to support or propose a hypothesis that each gene, we'll say one gene encodes one enzyme, and the enzymes are what's controlling metabolism in Neurospora crassa, in other cellular, in other types of cells, in other organisms that need metabolism, which is all organisms. Okay, so one gene encodes one enzyme. One enzyme, that doesn't make sense. One enzyme. And in the case of this fungus, this enzyme would be in charge of making leucine. So you mutate that gene, prevent the fungus from having a functional form of this enzyme. You prevent it from being able to make leucine. You need to provide it in the medium for the fungus to grow. So that's essentially how Beadle and Tatum's experiments worked. And they won the Nobel Prize for those experiments, and they isolated hundreds of auxotrophic mutants, each one requiring a different supplement to grow. Could be a vitamin, could be a nucleic acid, amino acid. Now, so what I wanted to talk about next was some experiments done by Serb and Horowitz. So Serb and Horowitz, who were also working with Neurospora crassa, and they extended Beadle and Tatum's work in a very interesting way. So now they isolated, let's say, some of the mutants they isolated were arginine mutants, or arginine auxotrophs. So in their mutants, Neurospora crassa, the mutants required arginine in the medium to grow. Arginine. It doesn't look right, right? Arginine. 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 Here we go. Let me write that again for you. Arginine. Okay, so what do they do? So let's say they mutate the fungus and they isolate one that requires arginine to grow. So they're going to call that mutant strain ARG1. And then they do the experiment again, and they isolate another arginine mutant. And they're also isolating mutants that can't grow without leucine, mutants that require methionine. They're finding all sorts of mutants, but they're finding arginine also over and over again. And when they find a new one, they give it a different number. So they do this experiment, and what they find is 7 independently isolated arginine mutants. All of these mutants right here, Neurospora crassa, isolated in different experiments. Because we really want to emphasize that these are independently isolated. So they're given different numbers, and each one of these isolates require arginine to grow on minimal medium. Well, minimal medium plus arginine. They can grow on that. They can't grow on regular minimal medium. Now, interestingly, so all grow on minimal medium plus arginine. Some grow on, I should be more prepared for that tip breaking when I grab a new pencil. Some grow on minimal medium plus a different compound plus citrulline. So all grow on arginine, but some also grow on minimal medium if you supplement the medium with citrulline. Then others, some grow on minimal medium plus ornithine. Okay, so what's going on here? So we can devise a table, of each strain and whether or not they grow on medium supplemented with arginine or citrulline or ornithine. And I think I have a table here. Let's see, where is it? Okay, so how would we organize this table? Let's see. Let's have the strain over here, and we're going to have a wild type strain as a control. We're going to have the ARG1 mutant, ARG2, and we're going to have all the mutants. 3, 4, 5, 6, 7. ARG, 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 ARG. Okay, so what if, we have minimal medium here, minimal medium with no supplement. Minimal medium plus no supplement. Now the wild type grows, but none of the others grow. Okay, so let's try minimal medium plus ornithine. Ornithine. Okay, wild type will grow. ARG1, the ARG1 mutant does not grow on minimal medium plus ornithine. ARG2 and ARG3, they do not grow. All the others grow when we add ornithine to the minimal medium. What about citrulline? So wild type grows. ARG1 does not grow. ARG2 and 3 both grow, and all of these grow also. And we already discussed minimal medium plus arginine. They all grow. So with the table, this is pretty interesting, right? Okay, so we can actually learn something about, you know, how arginine is being produced in the fungus from a table like this. Essentially, what Serb and Horowitz had uncovered was genes that are involved in making the enzymes that control a biosynthetic pathway. So biosynthetic pathway. So arginine isn't, you know, there isn't just one enzyme that, you know, takes very, very basic components and then presto, assembles them into arginine. So arginine is assembled through a complex pathway, and it's assembled in steps. And you go from a simple molecule all the way to the complex arginine. And each step in the pathway requires a different enzyme. So in class, if you remember, if you were in class, we went over a way that you can kind of try to propose a pathway to assemble arginine from data like this. And let me see if I can remember how we came up with what we came up with in class. I think I have it written down somewhere. I think we're going to rank the compounds and the genes. Let's see if this works. Okay, so what we're going to do is we are going to rank compounds by how often they allow a mutant to grow. And we're also going to rank genes, or rank mutations, or rank the mutants by least number of compounds that allow growth. Okay, rank compounds about how often they allow mutants to grow. We have 3 compounds, right? We have arginine, we have citrulline, and we have ornithine. Ornithine. There we go. So let's see. Now arginine allowed everything to grow, right? 1, 2, well, this is wild type, so we don't really need to include that, but 1, 2, 3, 4, 5, 6, 7. Okay, so arginine allowed 7 different mutants to grow. Citrulline, 1, 2, 3, 4, 5, 6, allowed 6 different mutants to grow. And ornithine allowed 1, 2, 3, 4 different mutants to grow. Okay, so how about the mutants? Arg1, 2, 3, 4, 5, 6, 7. So we're ranking the mutants by least number of compounds that allow growth. So let's see, let's just take Arg1 right here. Arg1, there was only 1 compound that allowed it to grow. So we're going to rank that guy number 1. We'll just say 1 compound first. How about Arg2? There were 2 compounds, citrulline and arginine. Same thing for Arg3. So let's go 2 compounds for these guys. And for 4, 5, 6, and 7, there were 3 different compounds that allowed growth. So let's go with 3 compounds for all of these guys. 3 compounds. So we have sort of a ranking system right here, where this is going to be number 1, this is going to be number 2, and this is going to be number 3. So I can come up with a biosynthetic pathway, you know, based on these. So now what's the easiest way to explain this? Let's go with ranking the mutants. Let's go with the compounds first. So arginine allowed all 7 to grow. So I'm going to put that at the very end. I'm going to assume arginine is the very last component in the, or compound in the biosynthetic pathway. And then I'm going to put citrulline next because it allows 6 to grow. And then ornithine only allowed 4. So I'm going to put ornithine here. Ornithine. Now, arginine, the ARG1 mutant here, only grew when it was supplied with arginine, only one compound. So I'm going to assume ARG1 is mutated in a gene, I'm going to say ARG1, that controls the conversion of citrulline to arginine. Now, ARG2 and 3, they required 2 compounds, so I'm going to guess that ARG2 and 3 are going to come somewhere down here, earlier in the pathway. This is our biosynthetic pathway, going from ornithine, going to citrulline, then to arginine. ARG2 and ARG3 probably come earlier in the pathway than ARG1 because there are multiple compounds that allow it to grow. And I'll explain more about this in a moment. And okay, ornithine can't come out of nowhere, right? So there's got to be a precursor, unknown precursor. So some enzymes got to control this step. Some enzymes got to control the production of ornithine from an unknown precursor. So I'm going to put all the others here because they all required 3 compounds, right? I mean, they all grew with all 3 compounds. So ARG4, 5, 6, and 7. Okay, so now we have a biosynthetic pathway, or at least this is our model of biosynthetic pathway. From this, not from this data, from this data, we came up with a ranking system that allowed us to come up with something like this. And then we would just need to check the biosynthetic pathway and make sure it makes sense with our model. So what is going on in the biosynthetic pathway? We're going from an unknown precursor. There's an enzyme being produced by ARG4, the ARG4 gene, and possibly the ARG5, ARG6, and ARG7 genes. More on this for a second. So one thing, we'll talk about this more. These could all be different mutations in the same gene, right? So let's say this gene right here is ornithine synthetase. You know, I don't know if that's a real enzyme name, but let's say it's the gene encoding ornithine synthetase. And the ARG4 mutant has a mutation right here. Okay, so ornithine synthetase, it's not functional anymore. The mutant cannot make ornithine. Let's say ARG5. ARG5 could be a mutation right here. So the ARG5 strain has a mutation right here, so it can no longer makes the enzyme that is required to synthesize ornithine. Same thing for all these guys. ARG6 could be here, ARG7 could be here. So that's one explanation for why all of these, ARG4, ARG5, ARG6, ARG7, why they're all, possibly at this step of the biosynthetic pathway, why they can all control this step. Maybe a better way to say that is why they are all unable to turn the unknown precursor into ornithine. So ARG2 and ARG3, what these would be in charge of, these are a gene that controls converting ornithine to citrulline, and ARG1 is in charge of controlling the conversion of citrulline to arginine. So if this is true, if this biosynthetic pathway is true, we should be able to provide citrulline in minimal medium to strains that are lacking ARG2 and ARG3. And citrulline should allow ARG2 and ARG3 mutants to grow. Let's say this arginine right here. Fungus requires this to grow. And an ARG2 and ARG3 mutant has a functional ARG1 gene. These mutants have a functional ARG1. So if we can provide these mutants with citrulline, well then those mutants can make arginine and they should be able to grow. So these mutants right here should be able to grow if we give it citrulline or arginine. Let's see at the table. ARG2 and ARG3, they do grow if we give citrulline and arginine. So they do grow. What about ornithine? Notice they don't grow when we add ornithine to the minimal medium. Well, that makes sense, right? So ARG2 and ARG3 mutants cannot convert ornithine to citrulline. So it doesn't matter how much ornithine we give these mutants, they cannot convert ornithine to citrulline. As a result, they cannot synthesize arginine and they're unable to grow on the medium. So we can check all of these, use that same sort of logical analysis process to make sure this table is consistent with all the steps in this proposed biosynthetic pathway. And we would find yes that this is consistent with this pathway. So what Serb and Horowitz did was they came up with a way to sort of figure out, not just the order of compounds in a biosynthetic pathway, how the compounds go from say the first precursors in the pathway to the final product of the pathway, in the order of those compounds in the pathway. So Serb and Horowitz came up with a genetic approach to sort of figure out the order of the compounds in a biosynthetic pathway, and which genes were involved in converting intermediates in the pathway to compounds further down in the pathway. So did they discover the genes yet? Well, they discovered mutations, and now after you discover a mutation that's in charge of, a mutation in an enzyme that's in charge of a certain part of the pathway, well, then you need to go find that gene and then characterize the mutation more closely. But finding the mutation first is the first step to finding the gene actually in the genome of the organism that encodes that enzyme that controls that step. So, so another thing, if we really understand what's going on, so if we were to grow, let's say, the ARG1 in medium supplemented with arginine, what intermediate would we expect to build up to high levels? If we had to pick one, which one would build up to high levels? So ARG1 mutant in minimal medium plus arginine. I always misspell that. Arginine. What compound would we expect to build up to high levels? Well, if I had to pick one, I would pick this one. Why? Well, because let's say the fungus is, its enzymes, all these enzymes are still working. This one is not. So we can expect that, the pathway is blocked right here. So the fungus should be building up high levels of this one right there, higher than normal. Because normally the fungus would be able to convert this to arginine. It can't do it anymore because this gene is mutated. So I would predict that this would be built up. Same thing when you grow any of these mutants. If you're adding supplements to the medium to allow the mutant to grow, you're adding supplements to bypass the part of the pathway in which the fungus is blocked. So you'd expect the molecule that is found right before the block in the pathway, we would expect that one to build up to higher levels, higher than normal. It might not be true. It might not be true that, as these levels start to rise, the pathway might be shut down. But that would be the first guess we would make, and it might be useful if, let's say, this was a very valuable compound, and you worked for a biotechnology company and you wanted to make lots and lots of this compound. Well, you could, theoretically use an ARG2 or ARG3 mutant, and you could have this compound build up to really high levels if you grew it in a medium that, you added citrulline or arginine. So you bypass the pathway, then the fungus is growing just fine, and it's building up high levels of this compound here. You do that with any mutant, theoretically, in the pathway. So that's it for Beadle and Tatum's experiments and Serb and Horowitz experiments, and in the next video we are going to take a look at translation. Okay, see you in the next video.